And I'll start by asking a question to get us warmed up. And this is to any of you. So when we've seen a number of cases of gene therapy being used. Last night we saw some really exciting examples of it being used to not replace a gene, but introduce a gene in humans. Obviously, there's been a lot of work done in mice also, and we've seen a couple examples now in cats. Are there other examples of products, gene therapy type products being developed for dogs or cats, the species that are of particular interest at this symposium?
Well, I think, one of the diseases that has also been worked on is probably one of the next products that gets approved by the FDA is for hemophilia, and I think it was brought up a little quickly yesterday. And as many veterinarians know, dogs have hemophilia as well, and many people have those animals and deal with the same issues that human patients do. And I think the dogs that were used to develop the human product, probably the longest are examples of the longest stable expression of a gene therapy product. So the two longest living dogs on that study actually passed away over the last 2 years at 13 and 14 years old. And they did not have one bleeding episode in that they were born, injected as young pups, not one bleeding episode in that entire period of time. And their factor IX levels were therapeutic that entire time. So I think that was a really good example for the potential for companion animal therapies as well as people, as well as example of the longevity that can be established for our goals here.
Linda Rhodes, I have a question about some of the data that you both presented. So David, you presented data on those 3 cats that showed 2 of them had elevated MIS levels, but the hormone data showed that they weren't cycling. So, and the hypothesis is that they're making antibodies against that MIS that in some way inactivates it. And Lindsay showed data that the GnRH antibodies were at a very low titer. So seemingly with the hypothesis that making anti-GnRH antibodies suppresses the expression level. So can you guys kind of reconcile those 2 things? So in one case, the treatment did not allow the overexpression of the transgene, and in the other case, the expression was there, but it wasn't active. So could you both kind of talk a little bit about that?
Yeah, so in my case, I actually had a mix of both mechanisms. So one of the animals, the levels dropped around the same time frame, 2 to 3 weeks. And I think this may represent a more active immune reaction. So maybe the difference between you and I is although 30 amino acid differences sounds like a lot, some are immunogenic and some are not. So I think it was just mildly immunogenic. Mm-hmm. And so in one case, there was a full immune response and the titer levels went down, which is typically what I'll see if I put a cross species, in a rat, for example. And then in the other cases, I think there's a milder immune response, so not enough to drop down the levels of the protein, but enough for whatever reason to interfere with the biological activity. So it's hard, but it's hard to know exactly what happened, but that's what I speculate.
Yeah, I was in it similarly. I think it depends on what part of the protein we're expressing, is what's stimulating the immune system. So perhaps with the GnRH antibody, it was enough to either bind it and clear it completely, and perhaps with MIS, it was just binding the part that interfered with the activity but still remained in circulation.
I think where we were maybe a little bit luckier is that actually we were well within the therapeutic levels. Mm-hmm. So you're still below, so we got to see the suppression for 60 days, although it was temporary.
Hi, Darren Noble from Ross University School of Veterinary Medicine. Thank you to both speakers. That was really interesting. I'm still trying to process it all, so forgive me if my questions are naive, but I'm going to ask them anyway. First of all, you guys are obviously cat people, so I was just wondering if anybody's looked at the sequence coverage for the dog MIS gene, and how likely is it to be able to kind of translate it into...
Yeah, so I didn't show this, but actually we do have the dog gene, and we've already cloned it, and we have a biologically active protein. So for all intents and purposes, we could go into dogs. So the dog genome project was more robust at the time. The only limitation is that most dogs are much larger than cats, so it's just production of the virus right now that's limiting.
Thank you. And just another question, if I may. I guess I've sort of always had the vision of, so my background is in rabies control, of having a product that you can inject in the field in the middle of a rabies campaign when they get pretty chaotic in some of these circumstances. And this isn't necessarily restricted to these kinds of products, but any permanent sterilant or injectable sterilant, we're going to have operator safety concerns and sort of human safety concerns. Is there any kind of, because people get exposed to the substance, they inject themselves accidentally or they get it in the eye or something like that. And obviously what you guys are doing now in a very controlled sort of circumstances, but is there any thought in the future to those kind of safety considerations? And what I guess I'm thinking about is species specificity, either of the AAV vector itself or of the protein in terms of the kind of species specificity there. And we've seen cats have maybe solved the problem for us by being a little bit out there, but any thoughts on that?
I think one of the important things to remember is that these are non-replicative, so it's very dose dependent, and we struggle to even, I think, get enough, as David just mentioned, to scale it up enough, for a cat. And so it's not a question of if you accidentally prick yourself that you're going to be contracepted the rest of your life. And so I think that's an important point to remember. And I think the approach depends as well. As we've seen in both of our cases, I think it's very species specific. And if you're creating a protein that's not right for that species, you're going to get an immune reaction. But of course, I think it's also, we can't dismiss all of these concerns altogether. And so it is something that we have to think about is how will you inject this safely? When we're doing these at Crew, we treat everything BSL level 2. Like our RBC says we're a little overly cautious, but we feel better doing it that way. And, one hand rule, you can't be holding with one hand. Our cats are anesthetized just to make everything as safe. We do it all in a hood. And we do that all over an overabundance of caution. So yeah, we obviously can't replicate that in the field, and we'll have to see what level of safety that everyone's kind of comfortable with when we get there.
Yeah, I can't stress this enough, because last time when I talked about this at ICSFR, I got a weird reaction. This will not be passed along from cat to cat. It's just it cannot work that way, because you need a massive dose and it's non-replicative, and you don't have to worry about it going from, I don't know, dog to fox or into the wildlife. And so for this to be a significant issue, you essentially have to inject yourself multiple doses, full doses. And even then, as I showed, the immune reaction would probably protect you. I still wouldn't encourage it, but I think it's relatively safe.
Just to add to that, as Linda mentioned, my primary role in the field over quite a few years was the toxicology studies, therefore establishing those safety profiles for these products, including vector shedding, especially depending on the route of injection. Case in point, we're now treating kids less than a year old, even younger, that are still nursing. So as you can imagine, that became an issue on whether that can be horizontally transmitted. And just exposure to saliva, any other bodily fluids. We've done a number of studies. It does get shed, but we've never had another animal get exposed enough that it even amounted to an immune response to the vector. As David and Lindsay mentioned, it takes really big doses to have any type of effect. So a lot of that work's been done. And when it comes time for these studies and others to go take that next step to demonstrate a safety profile, plenty of information is going to be out there in these target species already. But yeah, it is a concern, and a lot of people are going to want to talk about it.
Yeah, she was next.
No.
Hi, my name's Laura Stoker. I'm from the FDA. Lindsay, I really liked your presentation. The PowerPoint was excellent, easy to follow. I had a question for you. I know that you guys had felinize the antibody, the DNA that you were using for the FC portion, so that it would be less likely that you would have an immune response against it. But since you still ended up having an anti-GnRH immune response, what would be your next steps forward to trying to avoid that in the future?
Well, again, I will restate my disclaimer that my role in this is not actually designing the construct. I know Bruce Hay, speaking with him, he had some ideas, just more parts of it that he needed to change that we were sort of hoping that it wouldn't need to be that changed. I cannot tell you specifically the changes he's looking into and wants to do those, so I apologize.
Yeah, no, that's okay. Do you know if there was any of the antibodies that you used in that study to look at the anti-GnRH antibodies? Were they specific enough to know what it was binding to on your product, or was it just more of a general, we have an antibody response?
Yeah, I think it was more
Okay.
general. That's another good question that they did in their lab. And I know they struggled with actually trying to figure out how to actually do this assay to prove that. And I know they tried a couple different ways. I didn't present all the data, but I think they had 3 different antibodies they tried, and
Okay.
2 of them did bind and show an increase over time, and the third one didn't. But I don't know specifically like what regions or what, so I'm sorry.
No, I appreciate it. That was helpful. Thank you.
Hi, I'm Jessica Heckman. I'm a veterinary researcher and a science writer. And I guess my question is sort of a more general form of the one that was just asked, which is that given that the evading the immune response seems to be the really big problem for this line of research, are there thoughts maybe that we've learned from the human side about what kinds of proteins to try to form that are, I mean, I guess we don't know yet what Dr. Pepin's results are going to be when he takes his modified transgene and puts it into cats. But maybe, I mean, is it possible that trying to replicate endogenous hormones is going to be a better approach than antibodies, or do we know anything like that from the human side?
Well, so there's a lot of studies in all kinds of different model animals. Obviously, the cats and dogs, there's a little bit less work done, but I think we can extrapolate that if you're using an endogenous protein, usually you're fine, and you're fine in humans, and humans are relatively diverse. I think when you're using antibodies, it's a little bit harder because the interface with the epitope itself is non-self. So you're going to run into that problem over and over, and I don't know how you exactly protect against that.
Yeah, I think our challenge is always with dogs and cats is obviously human genome and the mouse genome are both, out there and well described, and people use it all the time. But cats and dogs, it's just a lot less information. So I think, as David pointed out in his talk as well, just getting that information and knowing what is the cat version of MIS, what is the cat version of this, is our challenge. And I think we just have to do that.
Although I'll say, as a dog genomicist, we're really threatened right now by the state of the cat genome, which apparently the most recent version is actually possibly better than the dog genome right now. Yeah, we're all quite upset about that in my laboratory. So, thanks.
I just want to expand on their answers a little bit. Their studies regarded antibodies and the immune responses that occurred to the antibodies. But we have learned quite a bit in the human side in regards to which genes. It's really a case-by-case, disease-by-disease basis. Alpha-1 antitrypsin, for example, the same gene can be used in mice all the way on up to people, whereas hexaminimose B, which results in Tay-Sachs disease, has to be mouse gene, cat gene, dog gene, primate gene, person, human gene. So it really is very much a case-by-case basis, and these 2 cases may differ on what they need to do what modifications need to be done.
Hi, I'm Giovanna Massei from Animal and Plant Health Agency. I'm trying to see the 3 of you. I have a question for David. Has anybody tried to do what you've done with the mice? That was very impressive, through oral delivery.
So I'm not aware of anybody delivering AAV through the oral route. There is intrapulmonary I've seen. There's brain injections, intramuscular, just IV, just anything but oral. I don't think it's possible with the technology right now. I've seen some interesting ways to deliver using not viruses, but protein through exosomes in milk, where it can bypass the gastric system and then get delivered systemically through the intestines. I don't know if that would maybe a way to bypass this.
To expand a little bit on that, some tongue injections and oral, just putting it in the mouth in the rodent water for diseases like Pompei's disease, where they lose the ability to talk because they lose the muscle wasting in their tongue. So they have Mai, who is at UMass as well, has actually tried just letting them drink the AAV, and it's transduced and it's expressed in the tongue and other gastrointestinal tissues. So it's the potential is there to eventually have an AAV vector that does do that better. It does it somewhat right now, but we're always trying to improve on that. So I know where you're headed, and it could be lead to very widespread applications as well.
Yeah,
it's just not anywhere as efficient, not nearly as efficient as an IM or an IV injection.
Okay. And a quick second question, if I may. Some of you mentioned the fact that it's even going to dog, you'll have to produce too much. So I assume it's still very expensive to produce this type of technology?
It is, especially at the proof of concept stage when you're doing studies that involve 3 or 6 large animals. There's quite economies of scale when it comes to gene therapy vectors. The type of preparations that are done by Guangping Gao for this study result in probably costs of somewhere around $2,000 per animal. But that's done in small tissue culture flasks. You need to do a lot of them, but there's companies right now, forming every month that are focusing on AAV vector production because there's a 3-year waiting list right now for clinical trials in some cases. But when you start moving up to 500 liter and 1,000 liter vats, large vats of preparations, the cost comes down tremendously for that same dose. So it's going to become a factor regardless down the road. It's just a matter of, it's really the limiting factor in AAV right now is producing enough. We could do plenty more experiments in clinical trials if we could make enough. So.
Good morning, Amy Fisher, University of Illinois. Thank you, that was very exciting. Very simple question. Is there a reason that you used the shoulder in one study and the quadriceps in another?
We actually didn't use the shoulder. It was actually the back leg for both. Yeah.
All right, that's the
difference between the shoulder and
I have a question for you, Tom. I'm going to put you on the spot.
Sure.
When I was doing lentivirus work earlier in my graduate studies, we could put ecotropic receptors so that we could do viruses that only infected mice and not humans. Is it possible to modify AAV in that way?
That's a good question. So that really comes down to the question is, could humans that come in contact with animals that are injected with a gene transfer sterilant potentially have an effect on themselves? I have not seen any work done on creating a species-specific AAV. There's plenty of work on tissue-specific AAVs, but not one that limits infection to humans and not dogs or cats or vice versa at this time. The possibilities of AAV, the capsid proteins that direct AAV to which tissue it goes to is the unlimited possibilities, and people are looking at that constantly to develop AAVs very specific to their use. So the potential's out there. You would have to, if you wanted to infect the liver in a human and not in a cat or vice versa, you'd have to find receptors that differ on the cat liver versus the human liver, and then add those specific ligands to the surface of the AAV. That would be most likely the way to do it. But I think livers are pretty close to being livers throughout mammals and so forth. Maybe.
Marc-Antoine Riencourt from France. Thanks to both speakers for very stimulating presentations. I have one question and one suggestion. The question relates to stability of your vector constructs. So you said it was fairly tedious to produce them, but if we contemplate using them in the long term, I guess you will need large amounts of this stuff to be stored at specific field sites. So the question is, how stable is the construct and how do you monitor and document stability?
AV is really stable, it turns out. In room temperature, some people have found it to be stable for months on end, but we prefer to keep it at minus 80, minus 60 to minus 80 degrees. In that case, there's vector that was used in the original, you guys saw Lancelot last night, vector that was used in that dog is still around and used as a standard for stability studies. So it's been aliquoted out in multiple vials in this freezer, and every once in a while, every 6 months or so, they take a vial out and measure it and see if it's still at the same potency, if they can do a potency test, but otherwise titer, which is done by PCR or a dot blot. But at minus 60, minus 80, it doesn't go anywhere. It's very, very stable. And I think it would be the same case for minus 20. So in regards to housing vector at various facilities in order to treat feral populations, as long as you had a well-controlled freezer, I think you'd be in good shape for quite a while.
Good.
Yeah.
And the suggestion is related to a possible other target. My background is sheep production. And in sheep, interfering with the GDF9 BMP15 pathway is leading to sterility exactly as shown by David with AMH. So have you ever considered embarking or sponsoring a project that would be dedicated to blocking also this GDF9 BMP15 pathway?
So I'm in my other life studying basically all the different hormones in the TGF-beta family. Sorry, I can't see you. It's kind of awkward. And the BMP and other TGF-beta family members are clearly important, not just in the early follicle growth, but even in primordial follicle, it turns out. And but it's a little bit more difficult to block something than just to overexpress it. So with MIS, we're lucky that just having high levels is enough to stop folliculogenesis. I'm not sure that would be the case for BMP15 or GDF9, but I would definitely think that blocking it with an antibody, for example, should work. But hopefully, as we tease out the different mechanisms of activation of primordial follicle, we'll find many other targets. And indeed, we found many new pathways that were previously not known to play a role in this. So I think it's going to open up another entire new class of contraceptive approaches, whereas you don't, instead of trying to disrupt the pituitary gonadotropin and gonad axis, you're actually going after the activation that's completely independent of cycle.
Hey, Matt Wilson, Scout Bio. We're actually a company focused on AAV delivery for companion animal health therapeutics. So quick question for both of the studies. Were neutralizing antibodies for the capsid taken prior to injection at all?
So we actually looked for it in vitro, so we did neutralization assays. There is a background neutralization that happens in cat serum in vitro for whatever reason, but we essentially saw no neutralizing ability in vivo. So we were basically the amount of virus we put gave us exactly the amount of transgene expression that we expected. So obviously it might change with exposure of those cats, especially in a wild situation. I don't know how that translates compared to husbandry. And so the contact with humans, I would think, would be it could be a limiting factor, since a lot of these are endemic. A lot of humans are endemically exposed to some of these AAVs. AV9 actually is the lowest, so we're somewhat safe with AV9. But AV2 is very highly, there's high titers, well, there's a high preponderance of titers against AAV2 in humans. So that maybe through shedding, the cats could be exposed.
Great. And just a follow-up question for you, David. Why the N-terminal sequence, or is it the N-terminal site in the MIS construct?
Oh, why keep it, you mean?
Yeah.
Actually, it doesn't fold and cleave properly if it's not present. So unfortunately, we can't just give the C-terminus. It's also, the N-terminus protects it in the circulation, so that it would have a very short half-life otherwise. So for multiple reasons of post-translational modifications, the N-terminus needs to be there, even though it's not technically binding to the receptor.
Great, thanks.
So I just want to make one more comment. One of the exciting things about the delivery of monoclonal GnRH antibody is that we know that in pretty much all mammalian species that have been tested, if you immunize against GnRH effectively, you can suppress reproduction. So we've known that in cows and in dogs and in cats. And so we know if you get a high enough titer of anti-GnRH antibodies, you can suppress. But the particular antibody that you chose, or that Bruce Hay chose, was fairly well characterized in terms of its binding. But in addition to getting serum levels, we're going to have an antibody that has a high affinity. I think that's why that particular antibody was chosen. So I guess one of the questions is, going forward, is it going to be important to better characterize the antibody prior to doing these studies for whatever species? Because we're actually giving a passive immunity. It's almost like collecting serum. In the old days, we collected serum from a horse and got the antibodies and used those to treat people for various conditions. And so we could characterize the binding. So it seems like it's going to have to be not only high titer, but a high affinity. So could you just talk a little bit about how kind of next steps have you talked to Bruce about? Is he looking at a range of antibodies for this? Because there's a variety of different ways you could change that.
Yeah, I know even before the study started that I always have trouble, SMI41, that does not flow off my tongue very well. I know he had looked at other anti-GnRH antibodies, and that seemed to have the best binding affinity in vivo, which is why he chose it. Yeah, and I absolutely agree with you. I think we were hoping we could sort of get away with this mouse monoclonal and just change a few things, and now we know that's not the case. So I do agree, and especially it's something we're going to have the cat expressing the rest of its life. It needs to not be immunogenic, because I think even if it's a little different, I think with time, we're going to run into the same issue again. We have talked to Bruce. I was hoping, we'd actually hope we'd be starting another one right now. I think funding right now is an issue for him. He has other projects, so I keep sort of bugging him every so often and checking in. And I know he has ideas on what he wants to change and has sort of the next step in his mind, but he hasn't started designing a construct. I think it's still, as of now, would be that same antibody, just more modifications, I believe.
Anyone else? We have a couple minutes left before the break. Any comments any of you want to make?
I thought a topic that comes up, and Matt already brought it up, is I mentioned in one of the talks yesterday that 90% of us in this room have antibodies to AAV2. Usually around that number. And then some of us have antibodies, a lower percentage to AAV5 and some of the other AAVs. It's just naturally occurring and infects us, doesn't cause any problems. But one of the things we need to consider in viral vector approaches is what kind of antibodies do dogs and cats already have. When we first started using dog models in these applications, we didn't give them parvovirus vaccines. It turns out AAV is a parvovirus. So no one knew the answer, but we had the concern. So we had them, very isolated, clean rooms at the vet school, so that parvovirus was not brought in by any of the students working with the animals and so forth. We know now that the antibodies or if a dog receives parvovirus vaccine, it does not interact with AAV2 or AAV8, AAV9, some of the popular vectors out there now. But I actually, this was a topic when we started our colonies. UPenn had the same questions, whether parvovirus vaccines was going to be an issue. It's not. There is potentially some cat vaccines that could also cause the same problems, but so far have not found any. I've done a few, when I was at UF, I did a few screening studies through the vet school there, just on samples, serum samples that came in as patients. We got approval, IACUC approval to do this, just screening cat and dog samples to the various AAVs. And they do have, both species do have a propensity to have antibodies against AAV8 or AAV9, some of the more popular ones. Nowhere are we going to find that there's an AAV that dogs or cats do not. Some of the animals do not have antibodies to. Even it may not even be from an AAV infection. It just could be an antibody to something else that happens to cross-react with AAV. So that already reduces us from having 100% of the population as available targets to maybe a little lower in some cases. But we are going to need to consider screening populations, feral populations, companion animal populations, to see their differences to the different AAV vectors or other viral vectors before we start treatments like this.
Although, from what I've read, intramuscular injection bypasses some of that pre-existing immunity.
Yeah, IM versus IV. And in fact, one of our grantees reported, and 2 other groups through personal communications have reported that cats that have antibodies to AAV9, it actually enhances their transduction of the AAV9 rather than inhibits it, interestingly. And it's been confirmed a few times now. So,
do you want to ask a quick? We just have a couple minutes for.
This is James Keeler with One Health Scientific. I appreciate Tom and Lindsay talking about pre-existing neutralizing antibodies to wild-type viruses or vaccines. One concern of elevated potential generating antibodies against an antibody might be to look at kidney function over time and clearance of the transgene antibody, because you could be eliciting a complement reaction and leaky kidney and fibrosis. So in the next trial, you might just pull urine and blood as well.
I should mention we do, during the study period, we are doing full physicals and doing blood draws. I think every, I can't remember if we did 3 months or 6 months for MIS, but we are checking those kind of values. So, but that's a good suggestion. We're not actually looking at urine and looking at kidney function, but that's a good suggestion. Thank you, James.
Well, please join me in thanking our panelists. Really exciting work.